No, she doesn't like me.
Hey everybody, welcome to Q&A with A&V. I'm Vincent Racaniello, and with me tonight is Amy Rosenfeld. Hello, Amy.
Hello, Vincent. No, she doesn't like me.
Always good. Hey everybody, welcome to Q&A with A&V. I'm Vincent Racaniello, and with me tonight is what do you got? Amy Rosenfeld. Hello, Amy.
I don't know. Hello, Vincent. No, she doesn't like me.
Always good. Hey everybody, welcome to Q&A with A&V. I'm Vincent Racaniello, and with me tonight is what do you got? Amy Rosenfeld. Hello, Amy.
I don't know. Hello, Vincent. No, she doesn't like me.
Always good. Hey everybody, welcome to Q&A with A&V. I'm Vincent Racaniello, and with me tonight is what do you got? Rosenfeld. Hello, Amy.
I don't know. Hello, Vincent. No, she doesn't like me.
Always good. Hey everybody, welcome to Q&A with A&V. I'm Vincent Racaniello, and with me try this. I'm switching my audio source. Is that better?
It's coming from you.
Is it better? That's what I want to know. Not if it's coming from me. Is it better?
Yeah, it's much better now.
Oi, oi, oi. Do I need a t-shirt with oi on it?
No.
Speaking of t-shirts, folks, here we go. This is what we got. 2 new t-shirts at cafepress.com/twiv. Life is an asymptote, either in white or black. And then we have, what's the other one? Right? No, that's the same one.
Why do you now? What's up
Amy?
with the picture?
What picture?
You're back to the home screen of Microbe TV Q&A with A&V. Hey.
Oh, right now it says right, Amy.
No, you pushed the wrong button.
Yeah, I do push wrong buttons. Anyway, this is also available in black, dark colors and white. Vanity Fair made the design, the the font and the colors and so forth. I need a producer, Amy. And I think we need a t-shirt. You need a t-shirt named that says "shocking," right?
Yeah, and then I need a vacation or a drink. Somebody just send wine. That would be good.
You could, send wine.
Just send wine.
Have a drink, Amy. In fact, maybe during these live streams, you should have a drink.
I can't bring alcohol to the lab. I'm still working. It's dangerous.
All right, folks. So you can get those t-shirts at cafepress.com/twiv. That's been up for a long time. There are a lot of cool t-shirts there. And Microbe TV makes $1 on each item. I don't put, I can mark up as much as I want, but I don't want to make them too expensive. You got sirens outside there, Amy?
Yeah, it's been a very siren-filled day today.
Susan wants to know what kind of wine you want.
Any kind will do at this point.
No, you don't want any kind.
No, I don't want any kind. I really like red or,
Pretty wants to know what's my favorite diagnostic platform. Amy.
Your favorite diagnostic platform is the peptide microarray by EN.
Is it? Why? That's your favorite?
Yeah, I think so.
I like
because the rest is just nucleic acid based, and it's just like a southern blot. This I think is more biologically relevant.
I like lateral flow assays. It's amazing what you can do. The simplest ones, they make a line on a paper sheet, but the more complicated ones actually make a QR code that you can read on a cell phone and transmit it, so you can read them in the field without any error in the transmission. So someone doesn't have to look at it and go, oh, it's positive or not positive. They just transmit the QR code. So cool, right? Right, Amy?
It's overkill. I'm not that. It's fine.
All right, Amy, what do you know about the first human case of H10N3 bird influenza?
I know it was a paper that I sent you that I read a couple of days ago.
Now, someone else
But
subsequently said it hasn't gone person to person yet.
I was going to say, I don't believe it has. I'm not sure that I'm all that. There seems to be a lot of spillover flu that doesn't seem to go anywhere.
Yes. By the way, folks, Amy, beforehand we had some technical issues, and Amy says, "Why do you have to futz with it?" But I'm a futzer at heart. I'm a tinkerer. I always tinker.
Yes.
Yesterday, I took apart the camera and I had to futz with it. So that's what happens. Okay.
You took apart the camera. You shouldn't tell me these things.
Well, I didn't
Well,
take it
clearly,
apart, but I put a new arm on it to make it easier to move around, because often when I'm on the TV show, they say, "Oh, push the camera back." Well, first they say push the laptop back, and I say I don't use a laptop. I use a proper camera, but I can't push it because it's locked in place. So now it's more flexible. Okay,
okay,
those are
sounds
mainly
good.
for the China TV shows.
Sounds good.
Why have vaccination rates dropped off in the U.S.? Do you know, Amy?
Um, well, so I think a lot of people have already been vaccinated that are in the age groups, and then I think what we're seeing are the people who are the hesitant, the wait and see what happens, the I don't trust the science kind of people. I think the people who are, the older people are like 70% vaccinated. So why? How many times can you get vaccinated? Now you have a ton of people who are hesitant to get vaccinated, and we
Yeah.
haven't done really a good
Yeah.
marketing of it because we've highlighted, if you're a premenopausal woman, don't get this vaccine, you know,
I
instead of,
also agree with Steph that low-hanging fruit has been taken, right? The people who want it are
well,
vaccinated.
that's what I'm saying.
Yeah. Here's another good one for us. In the RNA polymerase lecture, you said coronavirus RNA is being nested, which could lead to a lot of recombination. Did you mean some of the short plus RNA parts? Right. So the way the mRNAs are made is the polymerase copies a bit of the template and then moves to another part of the template, and it
Yeah,
joins
but that's
two.
not recombination.
Yes, but it's the template switch which can give rise to recombination, the movement of the polymerase.
But
So
that's not, no, recombination occurs during genome replication, not synthesis of the mRNA.
yes, I know, but the polymerase is very good at that kind of jumping, and that's why it does recombination well on the genome, because it's efficient at making mRNA. That's what is the thing there.
What?
The laws
Okay.
of physics mean you can't have 4 positively charged amino acids in a row. The only way you can get this is if you artificially manufacture it. Yeah, but this guy, Dalgleish, this is baloney, right, Amy?
What law of physics says you can't have 4 positively charged amino acids in a row?
The one that
Is,
Dalgleish made up and told the Daily Mail. There's no such thing. You can have many positively charged amino acids in a row. He's just hoping people
isn't a
are not
lysine
critical.
zipper a ring of positively charged amino acids?
Yeah, there are plenty of examples. RRAR, the furion cleavage site, is present in many proteins, not just SARS-CoV-2. This is just BS. This is just BS.
Well, it's present in hundreds of thousands of cellular
Yeah.
proteins.
Yep.
Because what does he think? Furin was just designed to cleave coronavirus spike proteins?
And why would anyone think to put a furin cleavage site in there? Who would do that? And it's ridiculous to even think about it. Anyway, they're all.
Well, I don't like to argue those kind of who would do something because
I
when
do.
I was a postdoc, no, when I was a postdoc, a very good friend of mine said to me, Americans are stupid. They thought you couldn't fly a plane, like you couldn't fly a plane into it. And I was like, no, I know you can fly a plane into it, but who would do it? And clearly there were 11 people who did it. So I don't like to argue these. Like, clearly there will be somebody who thinks to put a furin cleavage site into something to cause cleavage when you make recombinant proteins. So you could do it,
Okay,
but
fine. But anyway, you can find
that's
coronaviruses with the
right.
proline cleavage site, with the furin
That's
cleavage site. You can find even in bat SARS-CoV-2 like viruses, it's approaching it. So,
that's a better argument.
oi, oi. When I
Not
typed,
oi.
when I,
It's just
what?
it's not oi. It's just it's a better argument. It leaves you less open to somebody who thinks that the lab leak hypothesis is plausible
Yeah.
to not being a plausible argument anymore.
Can you comment on the supposed revelations from the Freedom of Information Act invoked to obtain Dr. Fauci's emails? Amy knows all about this, right?
I don't know. Can I comment about the supposed revelations?
Yeah, what are the revelations, Brian, that you want to know about?
I mean, I think it's kind of obscene that Morgan Fairchild and Mark Zuckerberg, who aren't scientists, get responses, but you and I, we have to go through channels.
Yeah, we do.
And we are the scientific community. So I think that that's a little obscene. But I don't know what I don't know what this whole question is.
Yeah, MERS coronavirus has a furin cleavage site, and one of the common cold coronas also does. Yes, you bet. Here's a question for Amy Rosenfeld. Is there PCR screening for B1.617.2?
Specifically for that? I would assume so, yeah. I would assume that there's a specific set of primers that covers that nucleotide change.
But if you got a fragment that spans it, I don't know where the fragment is, it would include them automatically. You wouldn't have to change the primers, right? Unless they happen to be
well,
in the
you
footprint.
have to do something that will discern it from the wild type. So either you have a
Yeah.
TACMAN probe, or nobody does what we used to do, which was restriction digesting, right? That's too long and too hard. Nobody would know how to do it today. So there has to be a TACMAN probe that either sits on top of it or something, or a change in size and band, but highly doubtful that there's a change in size and band.
Okay.
So there has to be something.
You think there'll be a rise in COVID cases since this past holiday, people traveling and getting together since the vaccine has waned?
What is the meaning of vaccine has waned? Is that the number of people who are getting vaccinated has waned, or is that the immunity induced by the vaccine? What does vaccine has waned mean?
I don't know, but if they mean antibodies, it doesn't matter if it wanes, because that's what memory cells are for.
Well, I don't think we've been vaccinating long enough for us to have antibodies wane. So
That's right.
I'm not concerned. If
Yep.
you're talking about the drop in percentage of people who are vaccinated, well, we're close to 40 some odd percent of people having fully been vaccinated, and over 60% of people who are half or partially vaccinated. So
Yeah.
I'm not really that concerned, and I don't really think that there'll be a rise in cases. And even if there were a rise in cases, I don't think there's going to be a corresponding rise in severe disease.
Exactly. What do you think of the WHO using Greek letters to label variants? Alpha, beta, delta.
I bet you Theodora has a really good perspective on it, because she's been tweeting about how unhappy she is. You
About
should talk to her.
the Greek
She's
letters?
great. Yes.
Okay.
Talk to her.
I think it is dumb, frankly. It means
I think
nothing.
that was kind of her response, but I'm sure that she will. I'm sure if you email her, she will write you a letter.
Okay. It is dumb because it tells you nothing. And I mean, the original nomenclature wasn't great, but at least people got used to it. And now we have to start over again. Well, bet you can guess what alpha is. That's easy. That's the 1.1.17.
Okay.
And
Well,
And then
whatever.
beta is 1.135, and then whatever.
It's it's yeah. I'm not learning this whole new system.
I don't think it's a good idea, but it's there. It's just like everything the WHO says. That's it. That's it. This is not quite related to COVID, but what makes some viruses almost always fatal when it reaches the brain, but not others like polio?
I don't know. You can check out Mateo's paper today.
You can't be cryptic, Amy. People want to know an answer.
Well, I didn't read Mateo's paper today, but he has a paper that they looked at virus from a kid who died of SSP, and they have a point mutation in the fusion protein, and they put it into systems that they learned from us, like organotypic brain slice cultures and organoids, and they say it's all about the F protein. So it's in JV online today.
So SSPE is a special case. Measles gets into the brain and causes encephalitis in about 1 in 1,000 measles infections. So it's not always fatal. It's fatal with SSPE, but that's a long-term sequelae, right? Rabies, yeah, when you get a dog bite and you haven't been vaccinated, virus will get into your brain, and 100% fatal. Yeah, we don't really understand. Polio is what is the fatality for bulbar polio? About 0.1% of all infections. And, the other forms of polio invasion are not as fatal. Even enterovirus D68, Amy's favorite virus, how often is it fatal? Very rarely, right?
Yeah. So really, we have no data on that, really. I mean, I think there have been 30 deaths, maybe less. But we don't even know where it is in those children who've died of it, because they're not subjected to autopsy. Like, we don't have a Bodian brain or a Bodian spinal cord. So there is one report waiting to be published about RNA being in the dorsal horn cells, but that's about it.
A portion of the medical community uses PCs versus Macs or Linux. I don't know about the medical community, but the science community is mainly Mac driven. If I give a lecture, well, in the old days when I stood in front of a class, most of the students had Macs, a few here and there had PCs. Meetings, when I go to science meetings, most people have Macs. Now the docs, I think they tend to have PCs because, their organizations are run by corporations, and corporations prefer PCs. But however, Daniel Griffin uses a Mac. I can tell you that. And so does Amy. You can see right behind her, quite an old Mac, actually. Probably not many people will recognize that one.
It is the old one.
Who said the WHO doesn't like Amy? Did you say that?
I don't know. I don't know. If I did, I don't remember.
How's the audio now? Are we good, or is it too
better.
loud? You're good? Better. How about someone just type in the chat? Is the audio okay? Let's give 10 seconds to get a response in the chat here. I shouldn't fool around. Is that what you're saying, Amy? Of course, the scientists use Macs. No, how's the audio?
Yeah, it's good.
I have that Mac Pro. Yeah, it's a Mac Pro. We don't use it anymore. Things don't run at it.
It wasn't really used. It was for running a microscope that we don't really know how to use.
Apparently the audio is good. And there's no more echo. It's all my fault. Yeah, I see now that we have... I don't know what happened. I had to switch the audio input, but I shouldn't be repeating. Nevertheless, let's get back to some questions. By the way, if you want to ask us a question, you could do at Vincent or at Amy, because I know you're having a conversation amongst yourselves, and it's great. And I'm just passing through all the audio issues. Okay, now here we go. Question. What's the criteria for assigning a novel virus to be studied at a specific B cell level? Do you want me to take this, Amy, or do you want to do it? Amy and I have arguments about this.
We don't have arguments about this.
So BSL-4, there you have viruses that for which there's no vaccine or any kind of therapeutic. It is high lethality and can transmit from person to person. So Ebola virus, person-to-person contact typically. Lassa virus. What else is a BSL-4? Nipah virus, Hendra virus. BSL-3, well, you're looking at a BSL-3 trained person right there in Dr. Rosenfeld.
I am.
Again, no vaccines or therapeutics and, somewhat lethal. Now, SARS-CoV-2 is a BSL-3, but I wonder if that's going to change once everyone is vaccinated. Know that you don't need a BSL-3 at that point anymore, right, Amy?
Right. I mean, it also all depends on who you talk to. Like, some members of the EHS community here at Columbia never thought that it should be a BSL-3.
Well, yeah. Who's that? Don't answer. You don't have to answer.
Well, she's not here anymore, so it's okay.
So does the biohazard can migrate around the room? Yeah, we have several of them, and when you empty them, you tend to migrate them and leave them where they are. But it gives a nice splash of color, no?
It is a good pop of color, and it happens that I was using the bench.
If you want to send Amy if you want to send Amy wine, I can give you her address. You could send it to the lab. It's fine.
Yeah, it could be. It's fine.
Okay, Mike wants to know, does the spike that comes from the vaccine not fold in the same way as the viral spike?
No, it folds in the same way.
Folds the same way. Now, the spike...
Could this lead to potential prion disease? Okay. So, Mike Frame, do you know what prion disease is?
Well, he can't answer you.
Well, okay. So I have a problem with this already. If you know what prion disease is, then you would never ask this question. Because prion disease is where an altered form of the protein seeds and causes a the correct form of the protein to refold and be altered again. There is no indication that when you fold spike, it is folding improperly. So how is this going to cause prion disease?
No, it won't cause prion disease.
It's not causing any other protein to... It's not causing any other protein. It's not causing any molecule of spike to fold improperly. So again, I don't understand. If you know what the disease is, why you would ask this question.
Well, he's just asking because he doesn't know, but the spike does not cause other spikes to misfold. That's the bottom line. Yeah, and speaking of spike, listen to this podcast where a DVM said the spike is actually a toxin and we made a huge mistake. That's completely absurd. It is not a toxin. It is a wonderful immunogen that provides immunity on its own. It is not toxic. This DVM, well, the DVM should go back to animals.
Well, first of all, I want to know why I would not make a vaccine out of a toxin, since I made a vaccine against diphtheria out of diphtheria toxin. It's very effective.
Yeah, you have to inactivate the toxin. Yeah.
Don't understand.
Yep.
I don't understand what the guy is talking about. If you know what DPT is, and isn't cholera toxin another vaccine? Don't we inactivate cholera toxin to generate cholera vaccine? So it is not clear to me that if you know anything about vaccinology, that we have not used toxins previously as immunogens in very potent, effective vaccines.
You need to go on Joe Rogan.
I don't need to go into work, and I don't need a sixth job. I already have too many jobs already.
It's just one evening. You go on and you shred them.
I just don't understand.
We don't actually have alcohol in the lab. No. I don't even think I have it in my office anymore. Used to.
No, we don't.
It's not prevented at a university. It's okay to have. But we don't have any more.
No.
So you could send Amy wine. And I don't know about the address you're talking about on the website. Let me give it to you, the correct address, okay? It's not hard. What's the furion cleavage site? Do you want to explain that, Amy?
Oh, my brain hurts.
All right, I'll do it. It's fine. Don't worry about it. Furins are proteases. Hubert,
I just don't get it.
proteases are enzymes that cut proteins, and they can recognize signals in the protein where to cut. Furins are a type of protease that are found in lots of cells, and we need them to live. And the furin site is what's recognized by these proteases. And SARS-CoV-2 has one, RRAR, right? 4 basic amino acids in a row, and it's cut by furins. And that's what all the brouhaha is about. Because some people say, oh, you can't have 4 basic amino acids in a row. That's nonsense. People are now making stuff up. Well, they've been making stuff up for years to see what they can get away with. And the press laps it up. Do you like bourbon, Amy? No, Amy likes, let me see. She likes vodka.
I do like vodka. I do like bourbon.
What kind of vodka do you like?
At this point, Eddie.
You like Stoli? No, you like
Usually I drink Kettle One.
Kettle One. Yeah, Amy likes bourbon too. Do you like the nice, single barrel bourbons, the handcrafted ones? Yeah.
I like a little Knob Hill.
Knob Hill is good, yeah. Is it possible for the vaccine to cause allergies to surface that weren't there before?
Ask Daniel.
Yeah, you can ask Daniel, but, I don't see why not. It can, the vaccine causes inflammation, and if you have an unbalanced immune system, it could trigger other reactions. So I don't see why not.
Okay.
Hey, Binax
Leonard the Great. I like this. Leonard the Great 33. Amy doesn't seem to in the right frame of mind to be live streaming.
She's fine. Don't give her hell. Binax now at Walmart for $20. Temper test. Very good. Antigen test. Are you going to get any?
He says he purchased several for the fall. Am I getting any? No.
Thoughts on the preprint out of Germany suggesting splicing errors in mRNA from the vector vaccines translating to soluble spike. I don't think that's the issue at all. I don't think that's it.
No, it's not. And I don't believe that there's any splicing in the mRNA because there's no splicing in the mRNA of the virus from the virus that encodes spike. It's not, So, no.
The corona polyprotein needs to be cut, but the protease that does it is part of the polyprotein. Well, one of them is. It's not
both of them are.
2 of them, but the furin is not. That's what I'm talking about. That's the spike, though. It's not the polyprotein, yeah.
Right, but the polyprotein as it gets produced is co-translationally proteolytically processed.
Yeah.
Just like a picorna is.
Yeah, this happens.
So they have a self-cleaving thing. They have a self-cleaving enzyme. No, it doesn't create a chicken or an egg problem.
It's not a problem. It happens in a lot of viruses. Listen to one of my lectures. The way to learn is to futz with things. That's right, Amy. You have to futz to learn.
I understand.
Latest news on the incubator. Monday, we are signing the lease, 9:30 a.m.
Excellent.
And then they have to do a little renovation. I guess it's going to take about a month. And then I go in and start to set up a studio.
Yeah, we give you till like July 13th.
Hey, look, he gets it right. Vinnie the Tinkerman. Love it. Love it. Maybe I should make a shirt for myself. Yeah, Tetris we forgot about because the Michael Mina lick-a-stick never happened. And we kind of lost steam after that. But, you know, these other antigen tests, $10,
It's very
it's too
expensive. It's
expensive.
way too expensive.
Maybe next year it'll come down in price. Maybe.
Well, next year you won't need it. Um, well, I mean, yeah, let's not talk about me now.
Can you explain why cold viruses are circulating among humans lately? Amy knows all about this.
So humans are the reservoir for a lot of the cold viruses like rhino and stuff. And so they, you know, when we came out of lockdown, you know, they just started circulating again. You got more exposure and you weren't immune anymore. Didn't we just answer this question for Carol?
Oh, last week, probably, yeah.
Well, Carol, she's in Israel with the grandkids. And
Yeah,
they
but
had
these people don't know Carol, so they
Yeah.
have to have it answered separately. It's fine. Israel has made a link to mRNA what? Vaccine in rare cases of myocarditis?
It's Pfizer and rare cases of myocarditis.
Mechanism unknown. Unknown. But why would Pfizer and not Moderna do it?
I'm not sure that they used that much Moderna when they vaccinated the majority of the Israeli population. I'm not sure.
Vincent, your room is dark. Well, that's a thing. That's the effect that I'm going for. It's got ambiance. I don't want it bright. Like Amy's got the bright lab, and I have the cozy little office here. What is spillover versus real transmission? Now, I know
Spillover is real transmission.
It's transmission, but
It's
spillover,
from one animal to the other animal is what we call spillover. And then from the same animal to the same animal, we call it transmission. But it's all actual transmission.
Yep. Peter, look at this is great. Thank you, Peter. Furin is a protein that cuts the spike. It allows the S2 spike to fuse with the cell and empty the virus into the cells. Beautifully said. Beautifully said.
Yes, that is what it does for this protein. But in general, it's an enzyme that cleaves lots of proteins.
How can it be determined if COVID was manipulated? Well, we look at the genome sequence, and it fits into a phylogenetic tree of other related coronaviruses perfectly. And it's not manipulated. And also listen to TWIV 762 with, I forgot the guy's name already. What was his name, our guest on Friday?
Gary.
Robert Gary. He tells you why it's completely natural. All the properties of the virus, including the receptor binding domain, is not something that is manufactured.
Well, I mean, the paper that you're doing on TWIV, they should listen to TWIV on Sunday.
Sure.
So the paper that you're doing on TWIV tells you it's not manufactured, that the receptor binding domain is not manufactured.
Yep.
And it tells you a great deal because they look at 27 different animals.
Mm-hmm.
And it's, you know, they do a lot of comparison, and they have kinetically they have a lot of different, it's all about, at the end of the day, it's all about kinetics, right?
It is. You took apart the camera. Actually, I didn't take it apart. I just pulled the wires out so I could put it on a new stand. My image is flat. Okay, so here's why it's flat.
What do you mean it's flat? It's three-dimensional.
It's a little
see depth.
Well, it's not as contrasty as usual, and the reason is there was a software update for the camera, and it erased the LUTs that I was using. LUTs is a lookup table that makes the colors what you want it to be, and I haven't got the LUT that I had before. That's why. I'll get it next week. Don't worry. It's really the content that matters, though, right?
Mm-hmm.
Someone asked
For
a
sure.
question about H10N3. Should we be losing sleep? No, not yet. By the way, this is my dog. Nice dog.
He's cute. I like the big floppy ears.
Could they make a malaria combination that fights the infective stage and also the bloodborne stage?
Talk to
Well,
our parasite people.
I don't see why not, because they got the infective vaccine. That's what we talked about on TWIP last time. Let's see. The Ontario government said, unlike the original strain, we know that some of the new variants we are fighting are more dangerous. Is there data to show this? No, Cat Castro. There are no data to show that. In fact, for the alpha strain or the alpha variant, it's not a strain. The alpha variant, the UK said it was more lethal, but it was a flawed analysis, and subsequent analysis showed that it's not more lethal. So until the paper comes out for this, what's going on in Ontario, no evidence for that. Are there any new but solid RCT data on ivermectin? No. We're going to have a guy on in a couple of weeks. No, next Tuesday, who's going to talk a little bit about that. In my country, the government is shortening the period between AstraZeneca vaccine doses. This seems counterintuitive, is it?
It's not what I would do.
What is the shortening? From what to what, Amy? Do you know?
I don't know because I don't understand what in my country is. So it's nondescript, number one. Number two, I wouldn't shorten, I would lengthen. But it just happens that my friend in Montreal who had gotten AstraZeneca, initially he said that he was not getting the second dose, I want to say until July 29th, because he was thinking about coming July 1st, or yeah, he was thinking about trying to come to the United States July 1st. But surprisingly, he got his second dose on Memorial Day, 8 weeks after his first dose. So maybe it's to, get everybody fully vaccinated who's partially vaccinated by the beginning of the summer, because it's hard to get people to come inside and remember to do it when it's so beautiful out and there's so many other things you'd like to do. Maybe that's the reason why. But scientifically, I would not argue to shorten the period of time.
Not less than what was done in the trials, 4 weeks, 3 to 4 weeks, right?
Yeah, I don't know what the time frame between doses for AstraZeneca was.
That's right. That might be different. Yep. How big a difference do you think full FDA approval of Moderna will make to the vaccine hesitant?
A significant number of differences. I think it will be significant.
Yeah, I agree. I think a lot of people are saying, oh, I'm not taking it because it's not approved. So,
yeah, they call it the experimental thing and we're not guinea pigs. And a few people have had some other analogies that I have rolled my eyes at, because I'm like, seriously, did you guys not understand what that historical event was? Because that was not it. But yeah, I think getting full FDA approval for both Moderna and Pfizer will eliminate a lot of problems. And then you can actually make it so that you have to be vaccinated or show vaccination, like when you go to public school.
So Jupiter, we don't know the answer to this, but Amy's theory is that, you thought that Moderna was less in Israel, right?
So I believe that the number of people who receive Moderna versus
the fraction.
Pfizer
Got it.
is less
Got it.
in Israel than other places. And that's why they've specifically highlighted that this myocarditis episode um, correlates with getting the Pfizer vaccine, because I think
Yeah.
like 3% of their population got Moderna or something.
Okay, got it.
Very less. But
Misunderstood.
the question is what is a prefusion spike? Why would a prefusion spike cause myocarditis?
Doesn't make sense because, yeah, the prefusion
Right,
spike will not cause fusion, right?
right.
Lab origins have been extensively discussed. New conspiracy theorists say the Chinese Communist Party is very powerful, got all the scientists to keep quiet for fear of death. Thoughts? Unlikely. I don't believe it.
I don't like to argue politics, and I don't like to argue or question people's integrity, because I wouldn't want mine questioned, especially by people who don't know me. So if you want to argue something scientific, that's a separate issue. Whether or not you want to argue politics and integrity, this is not, I'm not the person to discuss that with.
I would say that Amy is an example of someone who could not be intimidated by the Communist Party. That's why
That
I
is
answer
true.
that. See that?
That is true. That is true. I am blunt and honest to a fault, and many times you would like me not to be, but I am.
You talking to me?
Yeah, I think sometimes you would like me not to be so honest and blunt and
For
kind of
sure.
have a filter.
For sure.
That's just not happening, right?
Yep. It's not
Okay.
happening.
Right.
Is the myocarditis they are finding with Pfizer vaccine temporary or permanent?
You don't know yet. I mean, it's been reported for about a month. I mean, or it's been alluded to for about a month. But so I don't think anybody has any idea.
Someone in Ohio is a millionaire tonight, and a teen gets a full 4-year scholarship to a university for getting vaccinated.
That's great.
Good.
I think
Excellent.
that getting a full scholarship to college, to university, is great.
Would pre-fusion configuration cause more T-cell epitopes or post? I don't think people have looked at it. Have they, Amy?
I'm sure Shane and Alessandro have the numbers. I personally don't have the numbers off of the top of my head, but I'm sure that they know.
Thank you, Susan, for your contribution. Greatly appreciated. Can my kid, who's a few months shy of 12, get the vaccine?
Well, if he's a few months shy, just wait.
What's the difference? Will the furion site make to protein modelers? I think you said they would not have predicted it binding tightly. Does this send them back to the drawing board? No, it's the receptor binding domain. If you wanted to, so the RATG, the bat SARS-CoV-2 like virus, receptor binding domain is not high affinity. And if you wanted to make it better, you would have made it more like, you would make it different according to protein modeling. The SARS-CoV-2 solution you wouldn't have modeled as being a higher affinity. And Gary talked about that on Friday.
Yeah, it's 8:45. I have a few more questions, like 3.
Is it possible to get long COVID after getting vaccinated?
If you were to get infected, I would assume it's possible. It's not been reported.
Yeah, I haven't reported it yet. Isn't the best argument against deliberately lab-created viruses that they would need to be tested with lots of people?
No, that's not
sure.
the best argument. The best argument that against a deliberately lab-created pandemic-causing virus is actually surveying and identifying SARS-like viruses in animals that are geographically unrelated, which is what they did. So not only did Lympha have his paper, but the paper from the group, is it UCSF? New Mexico? What paper from the food from Nature Food did I send you on Sunday that they looked at like the viruses that are near the caves in China and Cambodia and Malaysia? You know the paper that
Mm-hmm.
I'm talking about?
Yeah, the one we're doing on Friday.
Is that the food paper?
No, it's not food. No.
I thought it was Nature Food where they sampled in the caves, and I sent it on Sunday.
Sorry, I don't recall.
All right, hold a second. Nature SARS, like viruses, land usage. You know the land usage paper.
Oh,
You know
yes, yes. Land use,
the land
ecological. Yeah,
usage.
yeah, yeah.
Yeah, the one that I sent
Mm,
you.
yeah.
It's the journal is called Nature Food, and it's called the Land Use Change and Livestock Revolution Increase the Risk of Zoonotic Transmission from the horseshoe bat, the rhinophaldi bat, right? And they look where all the bats are, and they look at they find bats that are, at the southern end of Russia and in Japan and Malaysia and all of that. That is the best argument, is that you can go and sample today and you could go and sample 10 years ago, and you can find these SARS-like viruses in these geographically divergent regions. And then you can calculate the distance that the bat travels, right? That's what Ian and Peter did in their NEPA paper, right?
Yep.
When they wanted to show that the NEPA belt and what is it, Kerala, the part in India that Dixon has gone to. You know what
Kerala.
I'm
Kerala.
Kerala. Remember that?
Mm-hmm. I do.
So then that's the best argument. The best argument is that these geographically divergent spots have very similar viruses and they
Yes.
have very similar bat populations. End of discussion. One more. My
Okay.
brain hurts.
Thank you for your super chat, K8. The good Dr. Osterholm made an appearance on Deadline White House. What on earth? Labs must improve safety measures. Why does he persist? It's his shtick. It's his shtick. Labs are. Ask Amy about BSL-3 security. She knows all about it. It's really safe.
Yeah.
Oh.
Nothing. Yeah. I don't want. Yeah. I'm not. He when he goes through BSL training and stuff, he can call and discuss it with me. But until that point, until he does lab work, until that point, I think he should not comment.
A lot of people think tuberculosis is gone, but I work with patients who have it active and latent and get treatment. I wonder if COVID is going to stay around in certain communities like TB has.
Yes. It's not a question. You have one more question, and then I have to go.
Do you think all quotes from scientists should include their specialty? For example, the new variant is extremely transmissible according to astrophysicist Jay Moon. It's great.
I do have to say I do love the fact that the majority of people who are commenting on transmissibility in virology are not virologists. They're social scientists, they're economists, they're bioinformaticians, there are various other people. But they are not the members of the virology community, and certainly not members of the coronavirology community, which I think, but why focus on the details?
One more.
One more.
Should healthy under 30s avoid adenoviral vector vaccines given that the risk of clotting seems to be higher than the risk of death by COVID?
The risk of clotting is lower than the risk by death by COVID.
Right.
The risk of clotting is actually lower than the risk of clotting from birth control pills. And healthy under 30, so unless you're a premenopausal male, which I've yet to meet. Have you met one yet, Vincent?
I have not.
I've yet to meet them. Yeah, I've yet to meet them. Uh, no, getting a vaccine is getting the Pfizer or not Pfizer, getting the J&J or AstraZeneca is fine.
Okay, Amy, thank you again.
All right, we need to change the CO2 tomorrow
Will
morning.
do. Will
All right,
do.
have a good evening.
Good night. Thank you, HZH, for your contribution. Are we sequencing enough to monitor the variants in the U.S.? No, we're not. But we're doing well, but it's still not enough. The world, no. I mean, many places are not doing any sequencing whatsoever. But that is a part of preparedness that we didn't do. Boy, the moderators are busy tonight. I'm sorry. That's where the thumbs downs are. A lot of thumbs downs tonight. We're attracting some trolls tonight who want to distract us. Why do COVID patients who have received monoclonals have to wait 90 days before being vaccinated? The monoclonals are against the spike protein. And the idea is you want to wait till the monoclonals clear out of your system so they will not bind to spike that is produced by the vaccine that you get, because the vaccines are making spike. That's the reason. I talk about thumbs down and I get more. You guys are great. Show about the Milwaukee Protocol. I think we did many years ago. We talked about, there was one young girl I remember in particular who was subjected to it. It's not a great idea. I think there's nothing else to do, but, it's not great. Is it bad that I want to keep my mask on until even more people are fully vaccinated? No, not at all. You can do what you want. I keep my mask on outdoors, and that's mainly because, I get out of my car and I walk into the building at Columbia. So what do I want to take it off for? If I go to the post office, I have to put it on when I go in, so I leave it on when I get out of the car. Is it safe to take a bus for an hour and a half with a mask if I've been vaccinated? Absolutely. Yeah, I mean, you're vaccinated, the worst is going to happen. You'll get a mild infection. But with the mask, you're not going to get infected. And we're having a thumb up, down battle. What a mature audience. Oh, boy. Okay, now the audio is good. Thank you. A lot of comments about that. Here we go. Get through that. Yeah, no, I'll fix the brightness and contrast. Don't worry. I told you, it's all about the LUT, the lookup table. You should dedicate an episode to making a presentation. Well, when I go to the studio, I'll make one. I don't think many people here would be interested in how you make the sausage, right? That's how they say it. What do you think about the Cancino vaccine? I think you should get what you were able to get. And yes, you could get Pfizer afterwards. There's absolutely no reason not to mix and match the vaccines. Ivermectin at symptom onset. A lot of people use it. I'm not going to say don't use it. I haven't seen the data. What we need is a large, which means 4,000 person randomized clinical trial to test it, because that hasn't been done yet. All the trials have been smaller and they've gone either way. So, but if someone uses it properly and they think it has an effect, let them do it. But as I said last time, if you're asking me, that's my thought. Should I ignore the CDC policies about unvaccinated people can take their mask off? I don't think you should take your mask off if you're not vaccinated. I don't think that's a good idea. I think vaccinated people can do it. It's up to them. But as we see, a lot of people here are choosing to keep it on. And that's fine. Macs don't have as many viruses as PCs, so I thought you'd like PCs. That's really good. That's really good. When I got my first computer at Columbia, well, actually my second one was a Mac. Mac had just come out. I was the only one at Columbia with a Mac. Why do doctors still use remdesivir? A lot of doctors run on inertia. They, heard something a while ago and they keep doing it, even though Daniel Griffin keeps saying it's not worth doing. Now in India, they've negotiated a price of $15 a dose, so they use it. I mean, there's probably not zero benefit, and it also would depend on when you give it to a patient. The later an infection, the less effect it's going to have. So, if it's going to save 2 out of 100 people, you could argue that that's worth it. But the monoclonals are really a better bet. SARS-1 caused more severe disease, and you ended up being hospitalized at the peak of transmission, and that's why it was easy to suppress transmission, because most of the people were hospitalized. Albertans who got AstraZeneca dose 1 are now offered the choice for dose 2 of either Pfizer or that. Well, so you want to know? Okay, I would get Pfizer, frankly, but in Israel, Pfizer was associated with myocarditis. So if you're worried about that, do the AstraZeneca. The side effects on the AstraZeneca are mostly on the first dose, so the second dose I don't think is a big issue. Can you explain this article published in Nature? SARS-CoV-2 infection induces long-lived bone marrow plasma cells. So this is what you would hope for in a vaccine, right? We would immunize you and you'd have memory cells, which are the bone marrow plasma cells. It's what you'd expect, and it's good news. So that means after vaccination, your blood antibody levels are going to decline to a low level, and then if you get infected again, the plasma cells are going to come out of the bone marrow and make antibody and interrupt the infection. So that's a good thing. It's really good. Amy likes red. Asking because he doesn't know is very generous. I tend to err on the side of generosity, right? I've been a teacher most of my life, and I always have this philosophy: there are no dumb questions. In my classroom, I want my students to ask me anything, and they do over the years. I get a reputation for that. Now, the live stream, my behavior bleeds over into the live stream, right? And I know it's a different beast, but it's hard to break habits, right? Hotez called for a full investigation of China. Does he watch your show? I'm sure he does not. Even though he's been on it, I'm sure he does not. I don't agree with him. Okay, let's see. I wonder why in vitro, is this Vincent, me, I suppose, in vitro experiments cannot be translated to human. Fauci downplayed Crati-Seti results. I wouldn't downplay. I would just say, look, these are results we get in an animal, and we have to test to see if they are working in people. That's it. You shouldn't downplay them. Maybe that's your interpretation of what he did. But it gives you a clue. Okay, we find this in an animal model. Let's look at it in humans. We can design the experiment to look at it. I think that's the value of an animal model. Remember, the vaccines are tested first in animals, right? If they completely predicted 100% of what happens in people, you wouldn't have to do clinical trials. You would just give them to people. But they don't. Sometimes the vaccine that works in an animal does not work in a person. So that's why they are models. Wow, we're just getting to the questions that I heard like 40 minutes ago. Oh my gosh. You have to understand that Amy has a full day behind her, right? She's there at 7 a.m. and then she's here 13 hours later and she's live streaming with us. I think it's great. How do you think protein-based vaccines like Novavax will compare? I think some of the preliminary data we saw looked pretty good for Novavax. It's an unusual configuration of the protein. It seems to be immunogenic. But, we don't have the data yet. It's too bad. I thought we would have it in May. How close does the corona have to be in the phylogenetic tree to be able to recombine in the same genus? Alpha coronas, beta, delta, gamma coronaviruses. I did ask Daniel Griffin if he'd do a live stream. He said he would. But he also said he would do it regularly, so he wanted to find the time. I just have to ask him. I have to get back to him. He's kind of busy right now. But things will ease up shortly, I'm sure, and he'll be able to do it. Why? So Amy's not here, Christy, but I'll take your question. Why do our bodies form correct antibodies to vaccine spikes when our bodies are exposed to all sides of the spike for a vaccine, but only the spiky end for the real live virus? Well, you make antibodies against all parts of all proteins, right? When you let's say a spike mRNA is injected into you, you're going to make spike protein. That spike protein is taken up by antigen-presenting cells. It's chopped up into little bits, and all the bits are displayed, and you get an immune response against many of them. But only certain antibodies are going to block infection in the case of spike. Don't worry about moderators, Vanity Nutrition, and Frank P. You're doing a great job. Don't apologize for anything. What do you see happening with the spread around South and Southeast Asia? Yeah, I think vaccination is the only answer. We have to help them. I mean, they're working hard to do it themselves, but the rest of the world needs to help, for sure. Is anyone doing serological surveys of blood banks to understand when things really caked here? Yes, absolutely. Probably it started earlier than we think. I'll bet when all the smoke settles, when the dust settles, we'll see that there were people infected in the U.S. maybe in December and even November, because travel is extensive between China and the U.S. Who else is signing? So me and Daniel Griffin are signing the lease. Actually, we're not. Our companies are signing, right? My company, Microbe TV, and his is Parasites Without Borders. So we are not personally liable. And will Brienne be at the studio regularly? I don't think she'll be regularly, but maybe a few times a year. She comes into New York. And someone asked, I think Vanity Nutrition asked, is there going to be a welcome party? Only for people who give a thumbs up. Yeah. It's a joke. Of course, listeners can come. I'm happy to answer any questions. But you don't have to be antagonistic. Do we know what types of animals are currently infected? No, we don't really. We know, cats, gorillas in the zoo, mink, ferrets. But how about rodents? I'll bet rodents are infected. So we had Tony Shounts on TWIV last year, who does field studies with rodents. And he said, a field study is you go out and you trap mice and you do something with them. So the potential for humans to infect them is real. So he wants to now go out in the spring and figure out if they're infected. I'll bet they will be. And by the way, Tony Shounts, next July, he has a bat meeting in Fort Collins, Colorado, and he invited me to do a TWIV there. So now we can start traveling again. I can start doing TWIVs. I used to do a lot of TWIVs on the road. Most of you probably didn't even know of them. But they were fun and really cool. We're going to start those again. So yes, we can have a studio warming party for vaccinated supporters. Yep. Can they really be $1 tests? I think in theory they could be. I know Min is an academic, but manufacturing scale, those are cheap pieces of paper that are printed. So I think eventually, but the current format is not, he envisioned a piece of paper like a pH paper that you would just put in your mouth and it would change color. But, a lateral flow assay is a little more complicated and it costs more. What would happen if the virus didn't have any spike, just a lipid ball with RNA in it? So that's a good question, right? Because the mRNA vaccines are basically lipid balls with mRNA in them, yet they appear to be able to get in cells. So the difference is the lipid nanoparticle mRNA vaccines have been designed to be able to be taken into cells, so their chemical composition facilitates their uptake. That was worked on for many years to get the right lipid composition to get into cells. And viruses can't do that kind of chemistry, so instead they have spike proteins in the membrane that helps them attach to cells and get in. So no virus with a membrane doesn't have a spike. It couldn't get into a cell. At least it wouldn't be very efficient. I appreciate Robert Gary's argument that the two lineages make lab leak less likely. I agree with him because you had to have two lineages establishing, and I think having multiple infected animals come into the market makes a lot of sense. Maybe two different markets, right? I mean, we know this happens for SARS-1. We know it happens, well, for MERS it's a different spillover, but for some of the other common cold coronas, it happened this way. Animals come into the meat market, the live meat markets in China. They come in alive. They're butchered there. If they have viruses, they're going to contaminate the people. In SARS-1, the people who had the highest seropositivity to SARS-1 were the meat handlers in the meat market because they were getting infected. And, we don't have that in this case. So we don't know to what extent, if any, the markets were involved. But they were epicenters early on, so it's kind of a hint. Anyway, I think that argument is a good argument. But you notice that the people who say it's a lab escape don't actually counter those. They just make other arguments that don't make any sense, like, the furion site is a smoking gun. Are there programs that determine the most recent common ancestor? Yes. Okay, so here's the story. Most of these programs, they depend on a good phylogenetic tree with lots of sampling, right? Lots of tips. And we don't have a good tree for the SARS-CoV-2-like viruses. It's pretty sparse. The branches are sparse. There is a new paper that came out in Molecular Biology and Evolution. We talked about it on TWEVO last week. They use a different approach to calculating the MRCA. They use a timing approach. They can take all of the sequences of human SARS-CoV-2 isolates. You know the date of sampling for each one. You know when the patient was swabbed, right? So now you can make a clock calculation based on that, and you can back calculate. And so they calculated the MRCA is late October, early November, which is earlier than anyone had said before. So in the case where your tree is sparse, you don't want to use the typical MRCA statistical prediction programs. Can you want to get the effects of long-term after being vaccinated? Not as the result of being vaccinated, but rather by getting infected after getting vaccinated. So I haven't seen it yet. Everything that happens gets reported pretty quickly. So I haven't seen it. I don't know, and that's a concern for people who don't want to get long COVID, right? I suspect it won't, but we'll see. Have you heard of virophages? Yes, I teach virophages in my virology course. Ask Vanity Nutrition. Yes, there's a virus. It doesn't actually infect mimivirus. It infects mimivirus-infected cells. And it's called Sputnik, right? It was named by the French, actually. 70% of adults in California have gotten their first dose. That's good. We're doing a pretty good job here. is Geertz's claims scientifically based? No, he doesn't have any claims for this idea that vaccinating is a bad idea because we're going to select variants, because infection will select variants as well. So what do you want to do? It doesn't make any sense. Here's a good list of animals: dogs, New World monkeys, felines, minks, otters, ferrets, pigs, primates, deer, mice, well, I don't know about the rodents in wild. That's the question I'd like to know. But yes, hamsters for sure, bats. Oh, yes, I wanted to mention the recent 212. So people said you have to address the lab origin. So I got members of the WHO committee, right? I got Robert Gary, who's written about it. I think they made a good case. And they explained it. Hypothetical question: if the current rhinovirus just appeared, we had not seen it before, would it have same tragic, deadly impact? Rhinoviruses tend not to be highly pathogenic in kids, the non-immune. So I don't think so. I think it's a different beast entirely. Yeah, there's a Vietnamese variant. Wherever you look, you can find variants. And you can give it a Greek name. What in a viral nucleotide sequence determine lineages? The homology. You take all your sequences of different viruses and you use a computer program to say which are the two most similar. And you pair them on a phylogenetic tree. You make those the tips of the tree, and then you go back to the roots. It's simply homology, that's all. Is it possible to elaborate on the family tree? What do you mean? Well, coronaviruses are all members of one family, which are very different from all these other viruses you list here. And you wouldn't even put them on the same tree because maybe only a small fraction of each genetic material would be similar. So we put similar viruses on phylogenetic trees because if you try and cross, say, virus families, the trees are not very good. Well, that's the mentality, or I should say the mindset of where we are. People like to scare people, I think. I don't know why. Maybe they want attention. But it's just multifactorial, right? The news media seems to amplify it. Here's Tyler. He says Canada's lying. They won't stop the fear-mongering. Is there any evidence for variant more transmissible? You know, there's circumstantial, but there's not direct proof. I don't think it's transmissibility. It's fitness. Because I can't imagine all these variants arising everywhere with huge R0 increases. It doesn't make any biological sense. I think the media are just doing what they're paid to do, right? They're just doing what they're paid to do. What are we going to do this fall with variants of concern and maybe the winter variants of high consequence? I think more people in the U.S., that's all I can speak to. Any country that's highly vaccinated, it's not going to be an issue. I don't think more and more variants are going to arise. Notice we see now the same amino acid changes and spike arising. Different combinations, but more or less the same ones. Any news of the Vietnam? No, it's just another variant. Probably shares some amino acid changes with others. But remember also, we're also focusing on the spike. These variants have changes elsewhere, but no one's paying any attention to them. It's all about the spike in antibody and vaccination efficiency. Does the ability of coronavirus polymerase to jump to form short plus parts an evolutionary trait? It certainly is an evolutionary trait, but the property is there so that it can make its messenger RNAs, which are made by a process of jumping. Want me to show you a slide to illustrate this? I'm going to get into my professorial mode as if I'm not in it already, right? Okay, let's see. Where would that be? Coronavirus. Yeah, I guess it would be in like a coronavirus lecture, right? So what's the last coronavirus? I have to go a COVID slide. Let's try that. I won't take too long here, folks. I want to get you this cool picture of how the RNA is made. Here you go. I got it. I actually found it. Okay, let's share this thingy. Here we go. And Vanity Nutrition will recognize this from the course. So here's the coronavirus genome in green, 30,000 bases. And from that are made all the mRNAs, which are overlapping. They're nested. The way they're made is that you copy a negative strand. So here's the negative strand down here in B. And you copy that into another plus strand. And then the polymerase starts here at the 3' end of the plus strand, starts to make the messenger RNAs. And then at some point, it jumps all the way to the 5' end and finishes copying. So that jumping is basically an ability to recombine, to do recombination, which the polymerase switches strands. And it does that to make all of these nested mRNAs. They have a common 5' end, and the 3' end is different. So that's what the jumping is all about that we're talking about. Get vaccinated, folks. If I get Moderna as my first vaccine, what should my second dose be? I think I answered this. You can get AstraZeneca. It's fine. It's going to prevent death and very serious disease. I don't think that's true, Michael. You can't require an experimental vaccine, but you might be able if it's fully approved. That's just false. I think Alan Dove has weighed in on that on Twiv. It's not correct. When will 11-year-olds, I think the goal is sometime this fall, they're going to do that. When do you expect herd immunity to be reached? You know, we're getting to the 50% overall immunity mark, right? Be combination of infections and vaccinations. So we need to get to 70%, but it's slowing down, right? And the infections are slowing down as well as the vaccination. So maybe not till next spring. It'll take a while. That's my favorite virus to study. Poliovirus. I've studied it my whole career. It's fascinating. I could talk for hours about it, and I have. I've given lectures on it. I heard the absence of nice camera of side effects in older people means their immune system is not as active. Does that mean they are somewhat less protected? So as you age, your immune system senesces, right? Your bone marrow shrinks. You can't make as many T cells. And yeah, your immune responses are poorer. And you have less inflammatory reactions to vaccines. And that's why with flu vaccines, old people need to get them very frequently because they don't make a good memory response. And in some cases, you need a higher dose, high-dose flu vaccine and shingles vaccine, for example. So you're absolutely right. Yes, Amy will not change. Do not worry. You know, I often say because she's very straightforward with me. And I say you need to filter yourself, but she won't. It's okay. What happened to UVC, far UVC? My understanding is some places have deployed it, and other people would know better than I about that. But yes, it's being deployed, but you don't see it typically because it's not going to be in a front-facing situation. I would love to have heard the job interview when Vincent hired Amy. Well, so she was my PhD student in the 90s, right? So it's not really a job interview. She came walking in. She said, I want to rotate in your lab and study this. I said, okay. And then she did great work, so I accepted her when she wanted to stay. Thank you, Ditching the Grind 3, for your contribution. We really appreciate it. Really appreciate it. If a survey or experiment is needed to prove natural spillover, how do you prove it? You find, it's just like SARS-1, Luis. In SARS-1, they looked for 13 years and they found civets had got a virus from bats, and they found a virus that was 99.9% identical to SARS-1. And that's what you need to do. Right now, we don't have 99.1% identity in any virus. All the viruses we have that are related are about 96% or less. It's not the ancestor. Don't let anyone tell you that someone was working with a SARS-CoV-2-like virus in the lab and it got out, because there are no such viruses known before December 2019. It's a fact. And if you want to tell me that they lied, well, then that's not science. That's something else. I got both doses of Pfizer. Then I got COVID. What does getting COVID after being vaccinated? So you must have gotten sick pretty close to the vaccine, right? But that's surprising because you should have been protected. But remember, there's a small fraction, 3% of people are not protected after vaccination. It doesn't do anything. You got a boost to your immunity. It's fine. As long as you recovered, I hope you have. Thank you, John Snyder, 2006, for your contribution. Really appreciate it. And by the way, folks, I forgot what I was going to say. Reading the comments and thinking is not optimal. Yeah, there are a lot of comments here. It's good. I love it. It's a nice community. That's why I scroll through to find the ones looking for to ask me, because you guys can talk among yourselves. But if you want to ask me a question, okay, you see, like, do add Vincent. That's why I can scroll through really quickly and get the ones that are for me. Vincent, what can you say about paleovirology sequencing ancient poop? Won't they just find viruses from bell peppers? All right, pretty. You're listening. I love it. They will, but they'll find other viruses too. I don't think people ate bell peppers forever. You know, but 15,000 years ago was the beginning of agriculture, right? So before that, probably not many people ate bell peppers. So you can find other things in poop. Osterholm is an epidemiologist, not a virologist. He should not publicly speculate about BSL-3. What about that the virus was a spillover? No, there's no evidence for that. There is no evidence. No one was working on anything close. And the two lineages circulating in Wuhan really argue against that. Thanks, Jack, for your contribution. Really appreciate it. I got J&J. Any chance I'll ever be able to get both doses of one of the mRNA vaccines? Yeah, sure, you can. You can just go get it. And, you know, if they decide we need boosters with different variant vaccines, you're going to get it anyway. Do you think domestic cats may have played any part in the transfer of SARS to humans? Cats eat bats. I don't know if they do. I'm sure if they found one, they would at least play with it, right? You know, the WHO committee, Marion Koopman said, not Koopman's, but in the report it says it could have been some other animal. It doesn't have to be a bat. So yeah, it could be something else, because a lot of animals can be infected with this virus. Triv 648 was also an episode discussing a computational biology on evolutionary origins. Yes, this virus, the SARS-CoV-2 like coronas, have been circulating unnoticed for decades. Nobody knew about these viruses. So to think that people were working on them in the lab is crazy. Nobody knew anything about him. I'm traveling with a friend who's not been vaccinated. I'm vaccinated. Should I make him wear a face mask? No, it's his or her choice, right? It's his or her choice what to do with their health. So you're good because you're vaccinated, and you're not going to be infected. You could wear a mask if you want. I don't think you should make him wear a mask. I would, if it were me. So I'm vaccinated and the friend is not. I would ask them to please wear a mask. Ask them nicely. Thumbs down. What is this? That Eric are good for the algorithm? Really? I thought thumbs down means that YouTube doesn't publicize it as much, right? Could be wrong. But Eric, you're the young guy. You might know. Is EVD68 really on the rise? Yeah, it is. But, this year was supposed to be an outbreak and we didn't have one, probably because of the distancing and masking and so forth. So, and the rise is not exponential, but it is going up. And kids are getting paralyzed and many don't recover. So it's an issue. They're all different issues, for sure. 926. Let's find the super chats and thank them. Thank you, X Jack Mac XX very much. Thank you so much. Got a lot of great questions here. Next time, put Vincent in front so I see them. Thank you, Michele, for your support. We really appreciate it. Please use the best questions from your Reddit to get started. Hey, there's great questions here. This is a good question, MD. Good thing you asked. I was waiting for someone to ask this. Why wouldn't a virus evolve not to rely on a spike for a fat-soluble blob? I think the fat-soluble blob works really well because we engineered it chemically. And it's really hard to engineer chemistry of membranes. It's much easier to put a protein in, and proteins are already in cells. So I think it's the lowest energy barrier solution. It's really good. It's a good question. Wendy, thank you for your contribution. Yeah. And post-polio, thank you again. We love your contributions. Have you heard of these weird infective particles called viruses? Yeah, I know. I heard something about that. Maybe I should look into it, right? Maybe I should look into it. Tero, thank you so much for your contributions. So, here on the live stream, I select the chat comments and they show up below me, right? The other thing I did is I put the camera too low. But the actual icons of the super chats don't show up, so they describe them. Pair character lifting some weights. It's funny because in the YouTube, you actually see the sticker, the super sticker, right? Is it okay to sign up for a booster clinical trial? Yeah. I mean, they may ask you if you've gotten fully vaccinated and then may say, no, you're not eligible, but you could try. Won't hurt. Thank you, Steph, from SF for your contribution. And Angela, thank you so much. How much protection should one shot give? Moderate and probably not long term. So eventually you need to get 2 shots. Here we go. HZH. Can virus be detected in stool? It can be, yes. And in fact, you can find it in waste sewage. It's an easy way to see if a virus is circulating in a certain area. And that's been done in many cities to look in sewage for a virus. Does that mean it reproduces in the GI tract? I think it's not been shown. I don't think anyone has shown in biopsies from human GI tract that the virus is reproducing. It could be passing through, right? Because you swallow your mucus. That's a way of getting rid of mucus. The cilia in your respiratory tract beat upwards. They bring mucus-containing viruses up, and you swallow them. That's how you get rid of them. So you always find respiratory viruses in feces. Whether the virus is reproducing there is another matter. Hard to detect. No one is really doing infectivity. You can do PCR. You find nucleic acid. Doesn't mean it's reproducing there. Thank you, Ian. Pleasure having your contribution. And that brings us to 9:30, folks. And another terrific live stream. A lot of down thumbs, but you know what? We got 237 ups. We answered a lot of questions. You got engaged. You had a nice little interaction here. You take what you can get, right, folks? So thanks for everyone for coming. Don't be disappointed in any questions I've skipped over. There are like 1,000 questions. I can't go through them all. I do my best. I do the science, right? Yeah, someone said, sorry, the ethics questions. I do the science. And thanks, everyone, for coming. We'll see you next week. We'll be here, same time, U.S. Eastern, 8 p.m. Thanks so much for joining us. And good night.